Good morning, afternoon, or evening, wherever time zone you are. In today's video, we are going to be talking about the structure and function of DNA. So we're just going to jump right into the video. DNA stands for deoxyribonucleic acid. It is a type of nucleic acid and one of the four major types of macromolecules, which are essential for life, along with proteins, lipids, and polysaccharides. Now, DNA is a double helix made of two single DNA strands. Each DNA strand is composed of many monomers called nucleotides. Especially as a result, each strand is a polynucleotide. Now, a nucleotide has three components: a nitrogen base, a phosphate group, and a 5-carbon sugar deoxyribonucleic. And these are sort of the DNA's backbone. The phosphate acts like a connector between the sugars by forming a phosphidase bonds between a third carbon atom and one sugar ring and the fifth carbon of the next sugar ring. These special carbon atoms are termed the 3' and 5'. As a result, there is a directionality to this backbone. Each strand has 5' end with a terminal phosphate group and a 3' end with a terminal phosphate group. Here is the phosphate group. Here it says from 3 to 5', and here is 3 to 5'. There are 4 nitrogen bases: adenine, guanine, thymine, and cytosine. Each of these attaches to a deoxyribonucleic via a glycosylic bond. The sequence of these 4 bases means that each DNA encodes genetic information, which can be used to be carried out of all cells' functions. Now, the central dogma of biology. The central dogma of biology describes how this genetic information is used by the cell. The DNA is subscribed into the RNA, which is then translated into the proteins. The four nitrogen bases can be divided into these two groups: pyrimids and purines. Now, the two single strands of DNA composing the DNA helix are held together via hydrogen bonds, and they run in anti-parallel in opposite direction, one strand going from 3 to 5, which is over here, while the other one is going from 5 to 3. Each nitrogen base on one strand binds to its complementary base pair on the other strand. Purines hydrogen bonds to pyrimines. Specifically, adenine bonds only to thymine via 2 hydrogen bonds. Cool. Alright, so thanks to this complementary binding that has been very beneficial, each of the 2 DNA strands contain the exact same information. When the 2 strands are separated during DNA replication, each of them acts as a template which contains all the information needed for creating the other ones. So it doesn't really matter, they all interchange. The 2 DNA strands can be melted in several different ways. They can either be through mechanical force, they can be through high temperatures, or they can be through extreme pH levels. Now, going back to the central dogma of biology, the DNA can get subscribed into the RNA, which then gets translated back into protein. However, though, not all sequences of DNA are actually meant to be transcribed. Now, I know that can be a little bit confusing. It was hard for me to grasp at first, but I hope I can make it just a little simpler for you. Those sequences of DNA which are actually meant to act as a template for transcription of RNA, which then gets translated into functional proteins called non-coding or antisense sequences. The DNA which is complementary to these antisense sequence is termed a coding or sense sequence. Remember, all transcribed RNA is complementary to the non-coding sequence, and so exactly matches the coding sequence. Both strands of DNA can have sense and antisense sequences. This means these sequences can overlap. For example, there can always be overlapping gems. There can be overlapping genes where one protein is encoded in another strand, while the opposite strand is read in the opposite direction. Since transcription can only proceed in the 3 to 5 direction template, the RNA being synthesized is always 5 to 3. Thus, this sequence, for example, will never be transcribed, although if the reverse sequence, it can be transcribed. The twisting of the double helix creates more of an interesting feature, in my opinion, actually. The grooves are not equally spaced, and the spaces, and one of these grooves is called the minor groove, and another one is called the major groove. Let me find that. Give me 1 second. Yeah, here it is. So these are the different grooves. We have a minor groove over here, and then we have a major groove over here. And basically what this do is really make the double helix so really more interesting. And yeah, in its relaxed state, in the relaxed state, each DNA sequence actually makes one loop around the axis every 10.4 base pairs. If the DNA is being twisted tighter in the same direction, it is called a positive supercoiling. If it is being twisted in the other direction, it will then be called a negative supercoiling. DNA is found in animals and plant cells. DNA is found in the nucleus in some eukaryotes and in the nucleotide in some prokaryotes. In both, though, the DNA is packaged into small chromosomes, which are combined of DNA and proteins. Chromosomes make these DNAs have more condensed and organized and allow the cell to have more controlled access to specific portions of protein. Eukaryotes have linear chromosomes, while prokaryotes have circular ones. The genome is a complete set of chromosomes in a cell. I hope you guys are able to understand a little simple lesson on the DNA, how they all interact with each other. I hope you guys have a wonderful rest of your day. See you guys soon.